Hi, this is part 2 of chapter 15, part 2 of 2. So we're going to talk about virulence factors. Virulence factors are molecules or things on top of, they're part of the cell of individual pathogens, and they are going to determine and help cause disease, and they can cause a very severe disease. A pathogen's virulent factors are encoded by the gene, so they're genetically, part of the microbe. They can include something called adhesins. We looked at that in the part 1, and also exoenzymes, exotoxins, and endotoxins. Adhesins are going to help a microbe bind to and remain attached to the host, right? So adhesins are going to be some kind of protein. Maybe it can be a glycoprotein, which is just a sugar on top of a protein that can be found on the surface of a pathogen and attaches to the host cell. So in this picture of E. coli, we can see that this red dot is going to represent a bacterial adhesin. The E. coli has a pili, right? These pili, remember, these are not flagella, but they are structures specifically to attach to the walls, to attach to a substrate, like something in the host. And that adhesin is going to be on the tip. And this adhesin, the shape of it is going to be able to bind to several different molecules on the surface of host cells, right? Host monocyte or host epithelial cell, doesn't matter, but this is just an example of what adhesins do. Adhesins are found on bacteria. They are found in viruses, fungi, protozoan. They just maintain the ability for the bacterium or whatever microbe it is to stay inside the host cell. There is a particular toxin, LPS. We learned about this when you learned about gram-negative cell walls. Lipopolysaccharide is considered an endotoxin. So what does endo mean? Endo is referring to the fact that it's part of the bacterial cell wall. It's part of the bacteria itself. It is not secreted out into the environment. Endotoxins can also be inside the cell, but it only becomes an issue when the endotoxin breaks free from the bacteria. So let's say that we have the bacteria is lysing. E. coli is a great example of our gram-negative bacteria. E. coli has LPS in its outer membrane. There is, remember, LPS, lipopolysaccharide. We have this lipid A part, and we have a very long O antigen. The lipid A part is actually very toxic to our bodies. And the O polysaccharide is something that we learn to label different forms of E. coli. Different strains of E. coli will have different O antigens. The reason why lipid A is so bad or gets toxic is because it acts as a super antigen, which we'll talk about soon. It causes inflammation and fever, but not just a little bit. It causes your immune system to be overwhelmed and uncontrolled. And so it really, huge high fever, a lot of inflammation. So that's the bad thing about lipid A. And since it's part of the E. coli cell wall, it's considered an endotoxin. However, it only is going to wreak havoc and promote inflammation and fever when it becomes loose. And so that happens when the E. coli lyse or die. So this is our endotoxin shocks. This is the probably the worst thing that could happen if you had a LPS and basically a gram-negative infection. You can see that the endotoxin here, right? So it can be other things too that can cause shock. But what shock is basically a rapid drop in blood pressure. This is that kind of shock caused by the endotoxin. And how does this toxin cause you to lose blood pressure? Well, if we look at, basically, we don't need to pay attention to all these small things, but we can just see that we're going into an immune cell, monocytes or macrophages. So this is representing an immune cell. This endotoxin causes the immune cell to really produce a bunch of different cytokines. So the word cytokine is going to be messages, chemical messengers between immune cells. So they talk to each other, communicate, and sort of coordinate an immune response with cytokines. When you produce too many cytokines, a lot of these cytokines will cause inflammation, causing your blood vessels to sort of dilate and open up. And then when that happens, I'll just draw a very rudimentary, this is a blood vessel. Okay, so here's your red blood cells going through your blood vessel. When you have when you have inflammation, you're right, and you have vasodilation, which is what's happening here. These are red blood cells. This is your tissue. Okay, this is not your blood vessel itself. This is your tissue. Okay, surrounding the blood vessel. So what could happen is that during inflammation, the cells that line your blood vessels get a little bit more opened up. The channels between them, they become a lot more permeable. And so a lot of the plasma, the water that's in your bloodstream, ends up leaving the blood vessel itself and sort of going into the tissues. And this is normally a good response. If you have an infection there, you want to recruit your white blood cells there, cause inflammation. But on a massive scale, on a systemic scale, it's very bad because all that water loss leads to hypotension, which is really low blood pressure. Basically, you have so little blood inside of your blood vessels that when your heart pumps it, it's not enough to reach your organs. So your brain, your lung, your kidney, all of those really important organs are not getting enough blood. And so you can see that here, and then that can lead to organ failure, and organ failure can lead to death, right? So endotoxin shock is this process, and LPS can cause this to happen. Now let's talk about some exotoxins. Exotoxins will be secreted from the blood, secreted into the tissue or into the bloodstream. So the bacteria makes it and then it's secreted. So before we move on to look at specific exotoxins, let's just take a generality of them. The endotoxin, we just talked about this. What is our endotoxin? Our endotoxin is LPS, okay? So it's only going to be found on gram-negative bacteria. For exotoxins, these are primarily going to be of gram-positive, but also gram-negative. So it's a little bit of both. Exotoxins are going to be proteins, okay? So they're going to be made of amino acids, whereas LPS, what is LPS? LPS is a lipopolysaccharide, right? So there's a lipid component and there's a sugar component, the polysaccharide, but not protein, okay? The effect on the host, you can have systemic symptoms, inflammation and fever. And for this exotoxins, they're going to be very specific. So each little toxin produced has sort of one target in the body. So they're very specific damage to cells dependent on the receptor-mediated target. So there's going to be a match between the exotoxin and some kind of molecule on the host cell, which is going to dictate the action of the toxin. Heat stability, LPS is heat stable. Whereas because proteins, right, we know proteins denature when the heat gets too hot. Most of them are heat labile, meaning that they're destroyed by heat. They're damaged with heat, but you do have some heat stable ones. LD50, remember the lethal dose 50 is high here and low here. So what does that mean? You need a lot of LPS in order to kill 50% of those infected with that number. The low means that you don't need as many exotoxins to cause 50% of those animals to die. So exotoxins are more virulent. They're not good, they're going to be the nastier one. Okay, so we're going to talk about some exoenzymes and toxins. Here are some terms: bacteremia, viremia, toxemia, and septicemia. So emia is what we have in common here, and that's in the blood. Okay, bacteria in the blood, viruses in the blood, toxins in the blood. Septicemia is a little bit different. Septicemia is bacteria are multiplying in the blood. So they're actually just reproducing in the bloodstream. Multiply. Okay. That means that that's actually very dangerous. The patients are considered septic at this point when they have septicemia, and it can also lead to septic shock, right? Just like exactly this kind of shock we talked about with LPS. And if we want to look at some exotoxins, they can cause disease by targeting the communication between cells, intracellular targeting. They can disrupt membranes, and they can act as superantigens. Superantigens, again, it's something that causes your immune system to overreact, and you can cause shock. So in looking at the first category here, so intracellular signaling, we're going to talk about botulinum and tetanus. So you want to know how these work. The botulinum toxin, right? This is our Botox again. So this is our connection point between a muscle cell and a neuron. The muscle cell is going to contract only when we have these neurotransmitters available in the cleft. So these neurotransmitters are called acetylcholine. The word is right here. And when acetylcholine is being released, and the word's right here too, when acetylcholine is being released, it binds to the receptors on the muscle cell, the muscle contracts. Well, Botox does not allow the release of acetylcholine, right? So there's blocks a mechanism here that drops the acetylcholine out. So the muscle cell never gets the message to contract. It is unable to contract, right? So that's going to be something we call flaccid paralysis. Your muscle cannot contract. It is flaccid, okay? And it's paralyzed, meaning that it's paralyzed in this like relaxed, flaccid form. The opposite toxin is tetanus, okay? Tetanus toxin. Tetanus toxin produces spastic, uncontrollable contraction. Okay, so here we again have this neuromuscular junction. And here we have an interesting thing. So if you've taken physiology, perhaps you've talked about this, but this is going to be an axon from one motor neuron, not the same one, a different one. And then this is going to modulate. This guy is going to tell this guy, hey, release acetylcholine, or don't release as much acetylcholine. So what tetanus does is that it prevents the release of glycine and GABA. So this is going to be found in the cleft. These are actually relaxing neurotransmitters. So it's kind of counterintuitive where like you have a lot of these released, but these tell this main guy here, no, the muscle needs to relax. So we're not going to spill out the acetylcholine. For tetanus takes this away. So it takes that no message, right? Don't contract. It takes it away so that this guy keeps continuously contracting. So the muscle keeps contracting abnormally because that no message is gone. And that's what tetanus does, okay? It prevents the release of glycine and GABA, which prevents relaxation. And for both of these toxins, because they work on skeletal muscle, your diaphragm is also skeletal muscle. So if your diaphragm is paralyzed, whether it's a flaccid paralysis or it's contracted and you can't relax it, you're not going to be able to breathe. And so that's how these toxins can kill you. Hemolysin. This is a particular toxin. We'll talk about this in lab, but it is something that destroys the membranes of cells. And in this case, hemo refers to red blood cells. Lyse is to lyse the red blood cells. So they are membrane disrupting toxins. Different microbes produce different kinds of hemolysins. But what we want to look at is this plate here that's completely red. This is what everything started off as. It's a red plate. And you can see there's some growth on this plate, but it didn't change the red color at all. So this is like basically no reaction, which we call gamma hemolysis, but don't worry about it right now. Don't worry about the terms here. We don't need the terms for this exam. I just want to show you the pictures. In this picture, we can see that there is a greenish color in where the red was, and that is because the toxin has punctured little holes in the membrane, causing the heme to come out, causing, and then the pigment changes because there's leakage, not total destruction. Here we have completely clear areas wherever the bacteria is. There's no red at all because in this case we have lysed completely the red blood cells. There are no more red blood cells where the bacteria grow because of the hemolysin produced. Okay, so hemolysin is going to disrupt red blood cell plasma membranes, and leukocidins, as you would guess, affects leukocytes, white blood cell plasma membranes. And now the superantigen, which we talked about before, molecules from microbes that trigger immune system to overreact, uncontrolled. LPS was our one example of a superantigen. Oh, I have it here. And excessive production of cytokines. Yeah, we make a cytokine storm. So remember that picture I showed you where you're going to have this, this cell, when you have this pathogen, if it's a superantigen, it causes your immune system to go nuts, and it causes a bunch of these cytokines to be secreted and released, and then it hits all these different organs, causing, massive drop in blood pressure. It's just a really messy condition. But that excessive production of cytokines is technically, you'll see this a lot actually, cytokine storm. It elicits a very extremely strong and